So now we're going to spend a few minutes talking about a couple of existing and ongoing prospective cohort studies within systemic JIA, and to think about the kinds of data that these are currently collecting and how they might inform some of these discussions we've been having, and how they could potentially be used as resources going forward. So I wonder, Bas, do you want to go first and talk about the Utrecht cohort? And then Yuki could talk more about the CARRA registry, and then I can end by talking specifically about the lung disease cohort as a sub-study within the registry.
Happy to, Grant, you're the chair. I think I'm going to share my screen then. Is it in presenting mode as well?
All good, thank you.
Yes, okay, looks great.
Cool. So I'd like to share some data and maybe an example of what could act as some kind of a control in some of the studies we envision, which is the Dutch experience. Actually, these are two cohorts. We'll be talking about patients that we have in Utrecht, so a single center from 2008 onwards, almost 14 years. But that evolved into a multi-center, actually national center in the 6 academic hospitals in the Netherlands from 2017 in an interventional trial, the ESTIS trial. It's an investigator-initiated trial of which I'm the PI. And the intervention is the taper and stop of anakinra, receptor IL-1 receptor antagonist, in those patients that achieve complete remission after 3 months. But of course, since this is an inception cohort, we also identified the 30% of patients that do not get into clinical inactive disease at that time point at least, and they are followed over 2 years. So although a small cohort, it's a multi-center cohort, and I think it may be of use of some of the things we are thinking of. The Utrecht cohort was started in 2008 by Dr. Nico Wulfraat, my boss actually. And in Utrecht, Holland is a small country, 17 million people. We see about 5 to 8 new patients per year, which makes up to more than 65 patients now. And they all have standardized follow-up and sampling since 2008. They are recently also all genotype with a full whole genome sequencing. And what is interesting of this particular cohort is that they all have been started on anakinra as a first-line drug without the start of corticosteroids. Of course, a significant number of patients needed corticosteroids next to anakinra if they did not achieve complete remission or switch to other registered biologicals. But the first line was really anakinra. From 2017, we mixed this up in, like I said, in an intervention trial. So we have available data of all of those Utrecht patients, but from 2017 also from the patients in the other centers. And at that time, we have clinical report forms at onset of disease, after 3 and 7 days, after 30 days, and at specific time points, quite often in the first year, and less often in the second year of the disease. We also have biosamplings of all of those time points, and particularly in those patients that taper and stop anakinra, we have even more time points, but that's only serum. And we have detailed clinical data, this disease activity data, add-on medications, information on adverse events or serious adverse events, and a range of patient-reported outcomes, including shock and Yamar. This is the treatment flow that we followed. So I will take you through all newly diagnosed systemic JIA patients. In the diagnostic workup, they were allowed to start NSAIDs, and at least for a couple of days to 1 week. If they had an insufficient response and they were diagnosed as systemic JIA, we started anakinra in a dose of 2 milligrams per kg per day with a maximum of 100 milligrams. If they still had fever at day 3, we increased the dose to a maximum of 4 milligrams per kg and a maximum of 200 milligrams a day. If not, if they responded by day 3 without fever, we continued for the anakinra for at least 3 months. We did biosampling and IL-18 levels at 2 months and 3 months. And if they had a full clinical response, at least ACR PET 90 or clinically inactive disease, and they have a biomarker which was IL-18 with a cutoff of 1200 picograms per milliliter, then we started a 1-month tapering, giving anakinra every other day instead of daily. And after 1 month, we did a full stop of anakinra. If they had clinically inactive disease but had IL-18 levels higher than 1200 picograms per ml, we continued anakinra on a daily basis and scheduled the patients for the next month assessment and did that again. And the interval of where tapering was possible had a maximum of 6 months. I will show you in the next slide. Of course, if they had at any time persistent or flares, persistent disease activity or flares, they were allowed to start concomitant therapy. Most of the time we started medium dose up to 0.5 milligrams per kg corticosteroids, sometimes with methotrexate, but most often without, or we switched to canakinumab or tocilizumab. And they were follow-up for at least 2 years, with a follow-up of at least every 3 months. So this is the data that was published on the Utrecht cohort, so not on the intervention trial, but on the Utrecht cohort in 2019. I think they are familiar to most of us. We had a pretty good response rate to anakinra first-line therapy, with 30 out of 42 in clinically inactive disease at 3 months. And at 12 months, also more than half of the patients in inactive disease or in clinical remission, actually, of medication. And here you can see that in the table, 75% at 1 year in clinically inactive disease, which extended to 3 and 5 years, although with lower patient numbers. And of course, also patients that switched to other drugs with either plus or minus corticosteroids, often also achieving clinically inactive disease. Of course, we also had patients with active disease, the numbers you can see here. These are patient-reported outcomes in the lower part of the table, and data on growth and on damage. And with this particular treatment flow, we minimize the use of corticosteroids, which also is displayed by very little damage or side effects of corticosteroids. Almost all of the patients had normal growth or catch-up growth, and also the weight was pretty good, actually, with the normal BMI index in most of the patients. The patient-reported outcomes were also quite okay. They are displayed here, with a maximum score in 2/3 of the patients. This is a nice graph of comparison of 12 patients who we are convinced have systemic JIA but did not show arthritis at onset of the disease, compared to 30 patients which fulfilled the EILA criteria. And you can see that the normal biochemistry lab, including some specialized immuno lab, soluble IL-2 receptor and IL-18, was not different between those. So this convinced us, or this is at least information that arthritis is not a required criterion for the diagnosis of systemic JIA. And very similar outcomes in both the patients with or without arthritis at onset. And as I said, this Utrecht experience then extended to a multi-center intervention trial. There's still inclusion needed. We are now at 45 patients out of 60 that are intended, so 90% inclusion. With a 2-year follow-up makes that we will probably end in 2024. The intervention part of the trial is only in the patients that respond completely to anakinra, and the intervention trial is power. We did a power calculation for that. Is intended to include 40 patients, which makes up around 60 to 65 patients that are included at onset of the disease. This is the follow-up scheme and also the measurement of the biomarker. So we have an open label lead-in phase. All of these patients are just started on anakinra, and only those patients that have a complete response at 3 months will enter then the intervention part, and then the tapering and stop of anakinra is based on the clinical response and the level of IL-18. And we measure that at time point 3 months, but also at time point 2 months to see how the kinetics of the IL-18 levels are, to be sure that the one-time measurement is not critical or at least we have an idea of the kinetics. I think it's also important to say that IL-18 was developed, actually, the assay in our lab, but since 2012, it's part of the diagnostic immunology department. So it's a regular and certified and also controlled measurement by our diagnostic department, which follows all the normal checks and controls. And our lab is doing that for all of the 6 academic centers. So the samples are transported to our lab, and we do this on a weekly basis. So also patients from the other centers have results of IL-18 levels within a week. It was funded by the Dutch Scientific Organization in a program for rational pharmacotherapy with the aim of making optimal use of expensive drugs like anakinra. But of course, this is a nice natural history cohort study as well. So to summarize, the Dutch Inception cohort and later the Estes intervention trial with anakinra therapy as first-line treatment is a high-quality data set of a standardized treatment protocol for systemic TIA, actually in a small nation in Europe. And I would like to ask and start a discussion of what steps can or need to be taken to use this data also as control data for future registration trials.
So, Bas, how many patients in this cohort would you qualify as refractory SJIA using the definition criteria that Fabrizio mentioned?
Yeah, so that's difficult. Alexi, important question. I think we have 10 or 11 episodes of MAS in the patients so far. So we don't see a reduction of the incidence of MAS occurring in our cohort using anakinra as first-line treatment. So that's for sure one of the potential subsets with refractory GIA. We have had only one patient with an isolated MAS in the liver. So if we have we talked about several subtypes or subsets of refractory disease that could fit, but we did not see interstitial lung disease in our cohort. Then, of course, we have a subset of patients that need corticosteroids to control their disease. So you could use you could define them as refractory, but definitely not like the cases that we encountered here today as from the patients and their parents with very refractory disease. It's a small cohort.
And in those patients, you did try other biologics other than anakinra.
Sure. Sure. So in the Netherlands, we have a national insurance program. So we have all of the patients have access to canakinumab, tocilizumab, and since last year also tofacitinib on a normal prescription base. So there's no limits to use these drugs.
Very nice, Bas. Thank you. I remember hearing first about the Estes trial actually at a SJIA Foundation meeting back in 2017 or 2018. And I'm very excited to hear some of the outcomes of that in the next couple of years. I had a couple of quick questions that I just jotted down. What in that sort of protocolized approach to sort of starting and escalating anakinra, what was the maximum dose you went up to before you sort of said that they failed that and sort of kicked them out, moving on to other alternative therapies?
So the study was actually aimed for to make optimal use of anakinra and also to stop it in those patients that are able to. So remember, and we defined it at a maximum dose of 200 milligrams a day or 4 milligrams per kg was a maximum as a single therapy only. So if patients needed more, and certainly we have some, certainly if they encountered MAS, then they were not allowed to go into the tapering and stop phase. But of course, we have the data on the follow-up of these patients, but they were not in the treatment protocol, and the doctors were allowed to treat as they thought was appropriate.
Makes sense. And then I think the laboratory data you showed on patients who did not have arthritis is very interesting. That basically sort of all of these sort of typical and emerging biomarkers that you looked at were pretty comparable. Did you have a set definition for how you defined or diagnosed them as systemic JIA in the absence of arthritis? Or any sort of a checklist of things that needed to be done for them as far as excluding other causes?
Yeah, so these 12 are from a single center, and these have been encountered in the period from 2008 till 2017. So you have to keep that in mind, only 12. And all of them got a full genetic screening. Most of them were also done a bone marrow punction, and I think 9 or 10 of them had a PET-CT to exclude malignancies and a full infectious screening, etc. So we were pretty sure that they had not another disease. Actually, a significant portion of them developed arthritis in the course of their disease. So they might not present it with arthritis, but they did later on.
Interesting. And I think Yuki will speak to that a little bit in a moment when she talks to the CARRA about the CARRA registry data as well. There's a question. So just again, restating the criteria for saying that they had a full response and then deciding to withdraw anakinra in the Estes, either in your original Utrecht cohort or the Estes trial.
Sorry, what was the question?
The criteria for withdrawing, yeah.
So either clinically inactive disease, so at least no fever, and 90 or 100% improvement of 3 of the 6 core set criteria. So actually clinically inactive disease.
Okay. Clinically active disease as sort of that sort of systemic modification of the Wallace criteria with fever as one of the criteria. Okay, very nice. Any additional questions specifically on this? And then I think we could talk more about this after we talk about what the CARRA registry has available. A long question there. What are you using when your patients do have high IL-18? So do you use that IL-18? I guess to sort of restate that a little bit. Does that IL-18 level, does that help inform when you pick other medications in those who aren't responding to sort of typical doses of anakinra, or have you primarily been using it at this point as sort of to help aid the withdrawal arm of this trial?
So in this trial, it's only helping the withdrawal of the drugs. Of course, we have data, and I can show one extra slide if you allow me, that tries to associate the IL-18 at onset of disease to the outcome of 1 year. And this is pretty rough data. I just calculated our single center data to the data that I have available now in Estes patients that have a 1-year follow-up. But that was not intended, was not powered for that. So here is the complete response. It's 70 patients now, and these are all IL-18 levels measured at onset of the disease, with a complete response or a non-complete response at 1 year. And non-complete does not necessarily mean that they are not in clinically inactive disease, but at least that they did not have a complete response to anakinra alone.
At that 200 milligrams or 4 per kilo threshold that you noted. Okay.
So you can see that the levels of IL-18 are somewhere between 10 to 20,000 at the median, but they don't seem to differ in the two cohorts.
Yeah, interesting.
And interesting, you see that, like I said yesterday, around 5% of the patients have low levels of IL-18, normal levels of IL-18. But that does not mean that they cannot respond to anakinra completely. And we have one systemic JIA with low levels that did not have a complete response.
And your lab's threshold for a normal is something in the 500 or so range?
Yeah, it's about that, 350, I believe.
Okay. Very nice. Okay, cool. Well, let's, why don't we...
Grant, I have a follow-up question.
Yes, please.
The reason I asked the criteria for stopping anakinra in this case is we've heard repeatedly that randomized withdrawal design is sort of not acceptable or not palatable, but what you guys are doing effectively is stopping an effective treatment. So I just wanted to kind of reconcile, this approach with this kind of reluctance to do randomized withdrawal designs.
Well, I mentioned that when I was discussing potential trials in MAS, which is sort of an acute event, not for other.
I think we've heard this concern also with trials in JIA. So I really want to have a good understanding so that we know how to approach these two.
Yeah. Bas, do you want to speak to that in your for your trial?
I'm not sure whether I completely get the question. Sorry, Nikolai.
Well, so what we have heard is that doing controlled trials, even in SJIA, it's sort of the broader indication, broad population is challenging, and prospective controlled trials are not acceptable because of the placebo. And randomized withdrawal trials were also, at least what we hear is that they're not really acceptable or considered acceptable by patients and I guess investigators. But what you guys are doing is essentially withdrawing a effective treatment.
Yeah, but I think this is a bit different because we know from this disease that this is a heterogeneous disease, which some patients, a significant subgroup has a monophasic course of the disease. And if you go to historical cohorts, that number of monophasic patients varies between around 30% to maybe 60%. And there also seems to be a window of opportunity to use IL-1 targeted treatment early in the disease. And there's quite compelling data on historic data that indeed confirms that at least a significant subset of patients is particularly sensitive to blocking IL-1 at the first, as a first treatment, actually, in the first phase of the disease. And this is exactly what we did in this trial. Not starting steroids, which, by the way, in 2008 was really the mainstream treatment of systemic JIA at that time. But we were interested in getting to know whether we use targeted IL-1 blocking therapy in all of the patients, allowing patients that did not get a complete response to get back to the then standardized treatment with corticosteroids, but the subset that did get into a clinical remission also allowed them to see whether they could get off of the therapy. And in fact, at least 50% of them at that time, without knowing the biomarker, was able to taper and stop in the first year. And looking back at that historical data, we found that those patients with high IL-8, or at least elevated IL-18 levels at onset of the disease, but had normalization of their levels within a couple of months, they were pretty sensitive to taper and stop the therapy. So I think that these are two different things. If I
agreed. Fabrizio has a comment on this as well, I believe.
Yeah, thank you, Grant, and thank you, Bas, for the reply. I think it's a completely different setting. If you are doing a standard withdrawal design in which you are randomizing patients who achieved an ACR 30 or a partial response, you have unstable patients who are being randomized to withdrawing an effective drug and their disease is not controlled. Here is a subset of patients which is doing great and they have achieved clinically inactive disease on drug, and you want to know whether there has been really a trained innate immunity, a training of innate immunity secondary to long-lasting remission. I see Bas nodding, and verifying whether this can be measured by IL-18 levels and allowing safe withdrawal of the drug because the patient doesn't need it anymore. And it's a completely different setting. And we certainly have the standard easy patients. That's not the ones we have been talking about, where the issue is withdrawing the treatment. It's not getting them to a response. It's withdrawing the treatment once we have achieved long-lasting remission. Now, the definition of long-lasting here is different. Biomarker guidance is different. And I mean, Bas has taken, if I may say, a very, I think one clear path chosen that one and is trying to investigate whether it works or not.
But we identify in this cohort of 65 patients, at least 30% of patients who are dependent on continuous maintenance therapy with canakinumab, tocilizumab, anakinra steroids. So you could define them as
non-responder to
non-responders to
anakinra
anakinra monotherapy.
Yes, not complete responder, non-responder. Here, the aim is very ambitious. It's always achieving clinically inactive disease. That you watch what we do in real life with the standard patients. Unfortunately, some of them are not standard.
Does that make sense and get at your questions?
Well, I think I was just curious about the very concept of randomized withdrawal, and the response before you randomize withdrawal patients could be defined differently. And I agree that ACR30 response is certainly far from remission, but you can use the higher bar of remission and randomize these patients. Again, I'm just trying to think about.
Then you have a sample size issue, Nikolai.
Well, it means that the available treatments are working pretty well, but that's not really
but that's
the case.
exactly the point of the discussion today. The available treatment are working pretty well in 80% of the patient, which is great. It's a new era, and the problem is knowing who can withdraw the treatment, who cannot. Okay, because they're too good. They're completely cured.
Yeah. Again, I just want to put it out on the table for the discussion that this approach should not be off the table and off limits for discussion.
No. But not certainly not in refractory patients. That would be off the table in refractory patients. For me as a clinician, I don't know about the others, but I will be very blunt in my reply.
I agree.
Yeah. That's sort of a different population. Although I guess at some point for these patients, once they're in complete remission, you would consider, managing their background treatments, right? Lowering background treatments.
No, these are patients in Bas trials are in monotherapy on anakinra, only anakinra only. So background treatment is not there. I think
it's a unique part of the disease where we know that many of these patients do have monophasic disease and will do well long term. And we think that we can quickly identify those who flare and recapture control fairly well, which doesn't necessarily translate to other types of JIA broadly, and certainly not other disease phases of systemic JIA. All right.
Not even to poly. You're right, Grant.
Correct. Exactly.
Yes.
Okay, so I now would like to transition a little bit and talk about the CARRA registry, and start with Yuki talking about the large cohort of systemic GIA patients who are in the CARRA registry and what we know about them, particularly as it relates to some of these things we've been discussing, like refractory disease and systemics who don't have arthritis.
Okay, so thank you for inviting me to speak about the registry and the kind of data that we have in the registry regarding systemic JIA that could potentially be used as historical or external controls for clinical trials. So I just want to give you a little background about the registry and how it started, and how enrollment is structured, as well as data collection. So the CARRA registry is a multi-purpose registry, which collects prospective data for multiple pediatric rheumatic diseases. And initially, the registry was constructed for phase 4 studies to satisfy post-marketing requirements. In terms of the enrollment criteria for systemic JIA, the ILAR criteria was not specifically required. We accepted physician pediatric rheumatologist diagnosis of systemic JIA as enough for enrollment in this category. And then the timeline of enrollment for systemic JIA varied a little bit. So basically all new, and new meaning patients who were diagnosed within 6 months of enrollment. and also previously diagnosed systemic JIA patients, meaning they could have been diagnosed years before, were allowed to be enrolled between June of 2015 when the registry started and January of 2020. Most recently, since the beginning of 2020, we've restricted the entry criteria a bit so that all new patients are still enrolled, but previously diagnosed systemic JIA patients are only enrolled if they start or restart a biologic or non-biologic DMARD. So the CARRA registry is 21 CFR Part 11 compliant database, which is managed by DCRI. So the data collection is very rigorous. And we collect all kinds of detailed clinical information, including medication start and stop dates, and also patient-reported outcomes, such as the CHAC, the JAMAR side effects questionnaire, and several PROMIS questionnaires as well. The data is collected about twice a year for most patients in the registry, but if they are enrolled in a sub-study in which more frequent data collection is needed, that is allowed, as you'll see in the FROST study. So as of February of 2022, we had 76 active sites and over 12,000 total patients enrolled in the registry, of whom 10,546 have JIA and 8% or 844 patients have systemic JIA. And this is taken from Peter's original presentation, which just shows the different cohorts of systemic JIA and their clinical manifestations. And you can see in the CARRA cohort as of this month, of the 844 patients, most of the characteristics are similar. And of these patients, 92% of them had arthritis at some point during their disease course. I have to say that the caveat with some of this data that I'm showing you today is that this was done relatively quickly and is not an in-depth analysis, so that more analysis is going to be needed to really refine the accuracy of this reported data, but it gives you a flavor of the types of patients that exist in the registry. So most of the patients, although not all the patients were enrolled at baseline, most of the patients were diagnosed within a fairly short timeline of diagnosis. So you can see 50% of the patients were enrolled within 2.9 months of diagnosis. But you can see that, as you know, there are patients who have very prolonged disease courses prior to enrollment, as long as more than 10 years in the maximum category. And in order to determine patients who did not have arthritis in this cohort, we looked for patients who had no arthritis reported at baseline, which could be any time during the disease course. And we found 90 or 10.6% of these patients had no arthritis. And then we went on to try to define systemic patients who never had arthritis and found that 64 of the 90 had 0 active joint counts at every visit in the registry. And of interest is 27 of them had visits with a physician global of greater than or equal to 3 without any active joints. So presumably that they had systemic only disease. We also went on to try to find potential refractory systemic JIA patients in the registry, and found that of the total 844, not just the patients without any report of arthritis, 13.5% of them, or 114, had an MD global of greater than or equal to 3 and a 0 joint count at least one visit. So they had at least one visit with disease activity without active joints. And we also looked for patients who have an indication of potentially being refractory because they were treated with both IL-1 and IL-6 inhibitors. And we found 190 of them that have been treated with both. And 140 of the 190 also had any use of steroid more than 12 months after initiating either an IL-1 or IL-6 inhibitor, implying again some resistance or refractoriness. And about 5% of the systemic JIA patients had at least one reported MAS event at or after enrollment. We do not have a good way of tracking MAS episodes prior to enrollment. We also capture SAEs and events of special interest, which are defined as adverse events that are of interest in the specific diseases that we study, but may not rise to the level of a serious adverse event. And these include adverse drug reactions, such as stress and lung disease, which we're now capturing in detail through Grant study, MAS, opportunistic infections, and liver disease are among the ones that we look at with interest in systemic JIA. I also just wanted to briefly mention a subcohort of the systemic JIA patients, which were enrolled in the FROST study. So this was an observational comparative effectiveness study of the systemic JIA consensus treatment plans using the CARRA registry. So these were new onset systemic JIA patients that were enrolled or tried to be enrolled prior to any treatment at all, and treated with one of the systemic JIA CTPs, which were with an IL-1 inhibitor, an IL-6 inhibitor, methotrexate, or steroids. And any of these non-steroid CTPs could also be given steroids along with the primary treatment. The primary outcome was clinically inactive disease off glucocorticoids at 9 months. And we enrolled 73 new onset patients. And in these patients, we collected data frequently at baseline, 2 weeks, 1 month, 3, 6, 9, and 12 months. And because the patients are enrolled in the registry, we continue to collect data on them at least twice a year for the next 10 years, which gives us an opportunity to see what happens to these patients and look at their disease course and treatment. So interestingly, although the study was intended to be, a comparative effectiveness study, the vast majority of the patients were treated with biologics because in comparison to when the CTPs were developed, which was in the early, 2010 to 2012, there were there was a lot less a treatment with biologics at that time. But by the time the study was funded, treatment practices had changed quite a bit. And so most of the patients were treated with biologics, and 90% of those treated with biologics were started on IL-1 inhibitors. And of the 10 patients who did not start a biologic CTP, 5 of them started a biologic within the first 3 months of treatment. So obviously we weren't able to do a comparative effectiveness study, but this still is a very valuable data set of systemic JIA disease at onset, documenting their early response with detailed clinical data. And we also have some home patient-reported outcomes as well, and detailed outcomes over a 12-month period, as well as biosamples that were collected at baseline and 6 months. So somewhat analogous to Bas's, initial cohort. And as I said, the follow-up will be able to be continued for at least another 10 years through the registry. So I think this is a very valuable data set and also biosample collection. I just wanted to point out that during the period of this study, actually, there were a total of 295 new systemic JIA patients that were enrolled in the registry during the period of time that FROST was active. So that actually there were, in addition to the 73 that were enrolled in FROST, 222 enrolled in the registry, also within 6 months of diagnosis, but did not enroll in FROST. And obviously there were many different reasons for not enrolling in FROST. But one interesting reason was that investigators repeatedly said that they wanted to treat their patients and did not want to wait for the arthritis to develop, even for that 1 week that was a requirement for entry in the FROST study. So I think that's interesting in terms of, studying patients that are new onset. This, having arthritis as a requirement is certainly going to be problematic because this may not develop early enough, and because investigators are not willing to wait for arthritis to develop. So I think both the CARRA registry and FROST data sets, contain, a rich amount of data that could potentially be used as either historical or contemporaneous real-world patient controls. And I think that, and of course, more detailed analyses can be done and are needed to identify specific phenotypes of patients that are desired as controls for specific trials. And I also wanted to say that, these controls can be identified and also matched to study patients using very rigorous entry criteria and sophisticated statistical methods. And we can also have, the ability to collect additional data if needed, as well as enroll additional patients to be followed prospectively with specific phenotypes. Most recently, the registry JIA patient data was approved by the PCORI methodology core to be used as a control arm for an interventional study for particular JIA. So to add to Bas's questions, I would say, under what circumstances would the FDA allow the use of such existing external data as a control arm for a registration trial? I think we got to speaking about a lot of this earlier on, and we need to do more investigating. And also, can the CARRA registry support the inclusion of Still's disease / systemic JIA patients that don't have arthritis in these registration trials, and whether information needs to be provided?
Yuki, I have a question on that point about the oligoarticular JIA. So that's very interesting, actually. So what parts of data did they feel could be a helpful historical control for oligoarticular JIA? Because that isn't an inception cohort like FROST is necessarily. I mean, some of the oligos in the CARRA registry are, but not all of them.
Well, I think, Laura is the PI of the study, so I think she could speak to that directly.
Yeah,
Where are
so
you on? Perfect.
you can hear me this time?
Yes, I can. Perfect.
Okay. So we just made the patients have to be an inception cohort.
Okay.
So even though the oligos within the registry aren't all an inception cohort, we only included those that were enrolled within the same time frame that was required for the clinical trial.
Okay.
So we were able, and so you go down from a couple thousand patients to, I'm trying to remember, I think 400 patients that were eligible for the trial. And then we also matched what kind of treatment they'd already, they couldn't have received any other kind of treatment, all the standard things you would expect.
This gets to that point from the earlier talk about saying when you use this, you have to make sure you're starting at the same point, right? And you can't just sort of take everybody with oligo JIA. You have to say, okay, let's match them up as best as we can to be an effective comparator to an intervention.
That's right. And we didn't take people that were in the registry for too long too. We only
Correct. Okay. So then to bring it back to the systemics, so certainly Frost, like the Dutch cohort, would be a comparator for new onset disease. The question is whether there are ways to tease out other subgroups within those systemics, those, as you were sort of alluding to, who have potentially refractory disease, whether it's sort of persistent steroid use for a period of time or have used multiple biologics.
Yuki, one question I wanted to ask, I should know that, but I don't remember. When a particular center decides to participate in your registry, how do they select patients to be in the registry? All the patients they have, consecutive patients diagnosed over a certain period of time, or at random? I guess the real question is there a chance that the registry is missing the more difficult patients that would have more data to enter, and the physicians would be avoiding enrolling that end of the spectrum in the registry? I think that's a real question.
Yeah, I mean, I think it's not that, every site is required to enroll every patient that they have. And there are some sites that enroll most of their patients, and other sites that, enroll only a fraction of the patients they see. And yes, there is obviously, there could be some selection bias in terms of enrolling patients that, don't have a complicated disease course, such as patients with lung disease, for example, because it just would, it would take a lot more effort to enter that data, which is why, one of the reasons why Grant and I, wanted to do this study to encourage people to enter those patients that exist that have lung disease. But yes, you're absolutely right. But but that's something also that could be, kind of improved by, if we had extra funding, for example, to enroll specific phenotypes of patients. That would be another way of enriching the registry cohort to, to have that. And the nice thing about the registry is that, there's uniform data collection. It's pretty thorough data collection. We collect, disease features, disease activity measures, multiple joint counts, physician, patient global, et cetera, and also PROs. So that would be very in a uniform way. And it would be easy to do. And we have 73 sites. So it would be.
May I make
I'm sorry.
I just wanted to comment a few things. First of all, I think if we analyze truly I don't know exactly how the arthritis was defined, but for example, my child is in the registry and we always wanted to be, but he does not have arthritis. To this day, I remember the day he was diagnosed, and the very well-meaning rheumatologist on hand said, "Oh, look at his second to left toe." And I was like, well, I don't see anything there, but they probably wanted to give him anakinra, so they diagnosed him. So I bet if we do analysis for who was actually presenting with arthritis on some kind of a cutoff there, we might find more these non-arthritis patients. So that's one thing. The second thing is that I think the patient-reported outcomes are completely arthritis focused. I mean, and that's everywhere. Yeah, I literally, any study I have participated in for SJIA, I stopped filling out the patient-reported outcomes as my small protest that this is not my child's problem. And I think a lot of the patients in the family group also will tell you the same thing, that they sit there and they are like, this is not my problem. Why am I, talking about, can he open the jar and all these things? So I think those we will need to, I think one of the things that's clear to me is that we need outcomes relevant to systemic patients and patient-reported outcomes and doctor-reported outcomes, all sorts of options if you had to go anywhere with this, because otherwise you are just measuring the wrong things for these patients.
Yeah, yeah. I think those are really important points, and I think I want to quickly present a couple slides on the lung disease patients in the registry, because we're kind of going to run up. I don't want to keep people past 1 o'clock. So let me do that quickly, and then we can have some more questions on the CARRA registry. All right, so this is just a quick update on this cohort within the registry. And so this was funded by a large grant from CARRA back in February 2020. Some stuff happened a little bit after February 2020. And so the original plan of it going for a year, we basically didn't even launch this in the first year, but it's now running initially through August of this year. Grant to myself, along with Yuki, Sherry Thornton here at Cincinnati, and Mark Natter at Boston. So the structure of the study was to get data on patients with systemic JIA and lung disease at 2 visits, a sort of a enrollment / initial visit, and then a 6-month follow-up visit. And at those visits, they would complete the normal registry data collections that you mentioned, those sort of biannual visits, but then have a supplementary data collection for some information, labs, medications that were more specific for lung disease that are not specifically asked about in the normal registry, along with getting a biosample at those 2 visits. And then sites were given an additional site payment for that supplemental data. Again, getting somewhat to the point that Alexi and Yuki had just talked about, that these are more complicated patients, and so to sort of give some more reimbursement to sites for getting this data. So this is a sub-study of the registry. So there's sort of 2 parts of eligibility. One is that they have to have systemic JIA, which, as Yuki mentioned, is not specified as meeting strictly the ILR criteria, but is a physician diagnosis. And so to try to capture some of those patients, because I know many patients who have lung disease are not, some of them are not formally called systemic JIA, they're called systemic JIA-like, but only because they didn't have arthritis, or they didn't have very impressive arthritis, and they didn't meet that ILR criteria, and that's really the only reason. And so we want those patients because we think they're the same phenotype. And again, these are not necessarily new onset systemic JIA patients, but by virtue of having the lung disease in this study, they're registry eligible for that. And then we came up with 3 definitions of lung disease because we have to sort of define this somehow. And we sort of looked at both clinical features, which can be things like tachypnea, cough, as well as clubbing, as well as things on chest imaging. And we said patients who have suspected lung disease have one of those. They have something on imaging but are completely asymptomatic, or they have clinical features such as clubbing but don't have anything clear on imaging. Most patients we think are in this probable bucket where they have both some amount of clinical symptoms that led them to get the imaging and then abnormalities in their imaging. And then groups in the last one have a tissue diagnosis. So some of these patients who've had a biopsy that confirm lung disease. So the additional information that we're collecting along with that, what's in the registry normally, at baseline, we ask some more information on their history of MAS, how many episodes they have, when they had them, whether they met the 2016 criteria, whether they'd been told they had subclinical MAS, clinical features of their lung disease, both at the time lung disease is detected and at the time of registry enrollment. And then information on the sort of the diagnostic imaging that led to that diagnosis, including features of their BAL, such as cell counts, any echocardiogram findings for pulmonary hypertension, CT findings, pulmonary function tests, as well as some other things that are specifically asked for. At both that baseline and then as well as the follow-up, we asked for something else that we invented called the Lung Disease Global Assessment, which is similar to that physician global assessment that Yuki mentioned on a 0 to 10 scale, because rheumatologists are used to filling that out, but that asks specifically about the lung disease, not asking about their arthritis. We're not asking about whether they're having a Still's rash. We're asking about their lung disease. We also ask about meds that we don't specifically ask about in the registry because they're not typically used for JIA, but are used for other kinds of interstitial lung disease, things like inhaled corticosteroids. And then some additional labs, including IL-18. So IL-18 is not one of the labs that is specifically queried by the registry. You can enter it, but as sort of a free text. But now we specifically query for that, as well as some other things like soluble IL-2 receptor, CXCL9, et cetera. And then also at the follow-up, we ask about change. And this is another thing that we think about as rheumatologists is, we say, since your last visit, are you much better, slightly better, the same, slightly worse, much worse? And again, we break it down into a couple different buckets because we don't want to focus just on these joints that are often not the big problem. We ask about their lung symptoms, we ask about their lung imaging, we ask about their SJIA symptoms, and then their SJIA labs, as well as asking about any new imaging, BAL, echocardiogram, or biopsies that occurred in the interim. And then finally, we are collecting biosamples. So CARRA has two standard biosample kits, one that is collected from more of those 73, 76 registry sites where they don't have access to a centrifuge tube. Those are non-spin kits, which really just gets DNA and then a Tempest tube for total blood RNA. But then a subset of sites do have the ability to spin these prior to shipment to the biobanks. We collect this larger group of samples, and this is what we're getting with this study, which allows us to get DNA, whole blood RNA, PBMCs, plasma, and serum. And we'll have those at baseline as well as at follow-up. And so we started enrollment. Our first patient was enrolled in July 2021. Since then, we've been enrolling about 1 to 2 patients a month at baseline, and we're now beginning to hit those follow-up visits from those initial patients. As of February 1, we had enrolled patients at 8 different sites and had, I believe, 13 patients enrolled with clinical data and 10 with baseline biosamples. So our goal, again, the official end of the funding that launched this is in the fall. We're hoping to have 20 initial visits here by the spring and something in the order of 30 to 40 by the fall. For the MDs on this site or on this call, if you are a CARRA registry site and you haven't heard from us about this study, shoot me an email. We would love to help get you launched. It's very easy. If you're a Reliant IRB site, there's just a memo you need to forward. If you're a local IRB site, we'll help you put through the amendment. If you're a patient who sees a rheumatologist in the US or Canada, ask them about the CARRA registry. If you aren't already in the CARRA registry, ask them if you can be in the CARRA registry and ask them about this sub-study. And then we're working on potentially other funding through CARRA as well as extramural sources to continue this collection, to get new patients in, as well as to continue to follow them long-term. And then with that, I am going to stop sharing so we can then, stop sharing. So then we can then spend these last few moments talking about other CARRA registry related questions. I see Bryce is asking, we are getting DNA from as many of these patients as possible. And the plan is we're likely going to, we are going to HLA type them. We don't have specific funds for that, but we're exploring collaborations with groups that do this, probably, including Mike Umbrello and the NIH group, to be able to have all of these patients genotyped and potentially have whole genome sequencing done on them all.
Yeah, and I think we're planning to do an RFA for use of these samples in the near future. So please be on the lookout for that announcement.
For both the frost samples and ones from this cohort.
Mm-hmm.
I'm looking up here at a couple, if there are other questions I missed while I was talking. Where is my cursor here? Yeah, again, sort of getting to questions about which patients are being enrolled at the CARRA sites. I think some of the ongoing analysis, Yuki, that you and Tim are doing on the Frost cohort, going to be looking at the patients who were enrolled versus the other registry patients who weren't enrolled to try to understand, what are the features of these patients, the generalizability of their responses, and then thinking for planning purposes, again, some of these barriers to enrollment, which I know we talked about a lot while Frosh was ongoing, about what can we do to try to get more patients enrolled at the time.
Yeah, well, it's a challenge to enroll, new onset patients of any kind, but especially systemic JIA patients, before treatment is very, very difficult. So.
My colleague Sue Thompson talks about the golden sample of that patient that can be enrolled in biosamples before they've seen any steroids or any DMARDs at that unique time that they're never going to have again. And CARRA has partnered with, through Melanie Kohlheim, to try to help working from the patient's side. She's a parent of a child with rheumatic disease, to help on the patient side with these translational research, because I think a lot of doctors, we will think, I don't want to bother a patient. They just got this new diagnosis. I don't want to bother them about a study or a biosample. They've already got a lot going on. And what we hear from Melanie and many other patients, including some I know on this call, is no, that's completely wrong. We want to help. We know, yes, we're overwhelmed. Yes, this is scary, but don't not talk to us about research because you think we can't handle it or we don't want to hear about it.
May I make a comment here?
Yeah.
And I'm going to sound like a broken record and a little bit, maybe not nice, but I'm going to say it anyway. Why do we need new onset patients? I think the whole field has been so focused on new onset patients and getting them and understanding their disease that we've just ignored the patients who are not new onset. They don't get enrolled in studies. And I mean, I think it's really remarkable how thorough the CARRA registry is and how much data there is and how much infrastructure there is. I am so jealous. And I'm so jealous because that's not going to help us right now, maybe in the future. And I think that if we are to truly focus on the entire subset population and look at where the unmet needs are, then we will need to change our focus from this new onset disease. Or either, I mean, from our perspective, we just go and build and do what we can separately. I'm sorry to put it so bluntly, but and there is such a dilemma in the new onset patients. We see this in the group. The new onset patients are not interested. They really are not. They are just, often they are passing by. They just get the disease, they move on. The drugs work well for them.
There are about 80 that do well.
And nobody asks us, except when we started saying, no, this is, so can we, because, and then, this is why maybe we need to just define ourselves as a new disease so that, I'm dead serious. I mean, if you truly ask everyone that, is this a new disease? Many of you will say, maybe it is. Maybe 10 years from now, we'll understand what the genetic basis is. Yeah, we are not sure. But we need to shift the focus of the field from the new onset.
Well,
the nice thing, and Yuki's probably going to say the exact same thing I'm doing, is that the registry infrastructure would allow us to do that. It's just having the money and the focus to sort of allow enrollment. So, for example, the lung disease cohort, these are by definition refractory patients.
Absolutely.
And we launched this when we wrote the initial CARRA grant to say, yes, this is not our registry target for enrollment, because a lot of the registry funding comes from, as Yuki mentioned, industry who want these patients when they start biologic for post-marketing surveillance, et cetera, which is great, but that's not this question. And so, finding funding to say we want to enroll refractory patients, we can do that within the registry infrastructure. There wouldn't have to be anything different doing with the registry consent. It would just be the funding to allow that to happen.
Maybe you could add the patients with liver disease, yeah? Then, maybe expand to what we discussed in the refractory patients. I completely agree. I mean, you have all the infrastructure, and in some sense, we are fighting the focus of industry, yeah? And because industry drives that new onset focus. And we are saying, no, go where the unmet needs are.
Well, I mean, we are not excluding patients, who don't have new onset, who've had disease. And actually for the first, 5 years of the registry, we enrolled all systemic JI patients who wanted to be enrolled or whose providers wanted to enroll them. And we are still enrolling systemic JI patients who are not new onset if they're starting a new medication, which happens a lot in refractory patients, right? So we're not actively excluding patients. We do want to study them. I think, there are two separate questions, right? That we're trying to answer when we do studies of new onset patients versus those that are refractory. And I totally agree with you. There's an unmet need here. And, if we can get the funding to enroll those patients, if industry is interested in studying these patients and doing clinical trials, perhaps the, answer, comes from that potential funding source, or there may be others that we can tap. But it is more labor intensive to enroll these patients. So I think that is a barrier. Also, if patients themselves went to their provider, and we have a list of all the CARRA registry sites, and they could go to this site and say, why am I not enrolled in the registry? They can certainly be enrolled that way as well.
Yeah, I mean, we can spread the word among the long-term patients who are actually more focused on the research. Anyway, I've probably,
No. But I think that's why we wanted to talk about these tools, to think about what are they currently doing and how can we use them to address the needs of these unmet needs. That's exactly the point of why we all wanted to be on there, talk about what's being collected.
True, makes sense.
It's like Nikolai and Fabrizio have questions.
Hey, Rashmi, I had more of a general comment, and I completely understand your frustration. That there is certainly a very clear unmet need in the more refractory patients. But I just want to caution that we should not be limiting the knowledge and the information that we get only to those patients, because there are other patients, the broader patient population, who may need a new drug, maybe not right now, but a year or two down the road, when they lose response to the currently available therapies. So, I certainly think that we have to think about the broader group as well.
Point taken. Yeah, point taken. I just think that when you build infrastructure and you build studies, you direct them both explicitly and implicitly towards certain questions. And, when we started off in the field, we were completely for just going with what was existing in the field. And CARRA is the best. There's no doubt about that, but, I think the studies have to be directed in a different way if they are to even capture. Otherwise, as I pointed out, we are the error bars, we get left behind in the error bars. So then, I think I've said my part.
Yep. Fabrizio, and then we're already running over. So I think maybe we'll have Fabrizio have the honor of the last question or comment. You're muted, my friend. There you go.
Shit. I think CARRA has the infrastructure. I believe it, and I'm sure it has the infrastructure. Just the point is the building. It's to change a little bit the variables they want to collect to make sure that they are relevant for increasing the knowledge and providing data that can be used in natural history study as a control arm. This is a must, and it should be done as soon as we can, using available infrastructure or not, and it's much easier if you have an available infrastructure. Yuki, I think you have a challenge ahead of you. One, just for a change.
I think we all do, not just.
Yeah, I think we all do.
For a change, right?
For a change.
Okay. Well, thank you so much to everybody from around the world for coming in here. Rashmi, I'll turn things last to you for to close out the day.
Yes, thank you, everyone. We are, glad to see so many of you still here. It's been a long day. Tomorrow we start off with some working sessions for the Accelerate Research Study and about discussing, I think, more of the natural history study. And also in the second half, we have an exciting session with some talks on SJIA lung disease and then a patient panel. to kind of really go for helping, I think, the physicians and everyone else understand what we are really seeing in the patient groups. So with that, thank you so much, and I will see everyone tomorrow.